Happy snow day and welcome to the answer to the do now. So I'm going to go over the do now and give you the answers to make sure that you got them right. So this is the do now. So it says place the enzymes below in the order they function and say what they do. So the first one that function is DNA helicase. And DNA helicase unwinds the two strands of DNA by breaking the hydrogen bonds between them. Then the unwound strand is likely to close back into each other because the nitrogen bases are attracted to each other. So the single-stranded binding protein, these guys, then hold the DNA open. Now, then it is the job of RNA primase to add a short RNA primer of about 8 to 10 bases. The reason that has to happen, we'll answer down here. Then DNA polymerase 3, at least on the leading strand, will synthesize the rest of the DNA until the end of the DNA. And then DNA polymerase 1 will remove the RNA primer. And on the leading strand, remember, it can't be replaced, so that causes the DNA to shorten. Now, DNA ligase is only required on the lagging strand. Remember, that's because the lagging strand has to be synthesized in Okazaki fragments, which we're going to talk about down here. And it is the job of DNA ligase to seal or bond together the Okazaki fragments. It basically bonds the sugar to the phosphate group on the Okazaki fragments. So, because of the way evolution solve this problem of DNA replication, there are some things that are not perfect about it. And one of the things that's not perfect about it is that DNA polymerase can only add nucleotides to an existing strand. So it has to have something to add on to, it has to have a sugar to add a phosphate group to. So we solve this problem by starting DNA replication with an RNA primer, so that DNA polymerase has a 3' carbon on a sugar that it can add to. So B is the problem that's the hardest to understand, and when I said in class I didn't really maybe understand it until I became a teacher. And Thursday in class we're going to try to model this to make B's problem make more sense. The problem with B is that DNA polymerase can only add nucleotides in the 5' to 3' direction. So that means that DNA polymerase on each strand has to go in opposite directions, okay? So, but the problem with that is that the fork only opens in one direction. So the way we solve that is one strand, so like the bottom strand, if we go to a slide, hold on one second. So, well, if you see my lovely drawing here that I just made using the mouse. So if we were going to find out which one was the leading and which one was the lagging strand, remember DNA synthesis has to synthesize in a 5' to 3' direction. So it uses the template. It starts at the 3' end of the template and goes this way. And this can just replicate and replicate and replicate and replicate and replicate and replicate. And I know I'm not drawing straight. And it's going to continue to replicate because this fork is opening in this direction. Now on this side, this is going to be the lagging strand. Remember, because it adds to the 3' end of the existing strand and goes to in the 5' and 3' direction, and it's going to continue going until it reaches the end here. But then what's going to happen is that this side is going to open. And so when this side opens, so this will open up, this here will open up, it's going to have to go back and replicate this area in using Okazaki fragments. So basically, to solve the last problem, DNA synthesis happens bidirectionally, and it has a lagging strand, which replicates the DNA strands using fragments called Okazaki fragments. Okay, I will see you tomorrow, and well, probably not, well, Thursday, but have a good snow day.